Good afternoon. My name is Sarah, and I will be your conference operator today. At this time, I would like to welcome everyone to the long-term effects of prenatal substance exposure on children conference call. All lines have been placed on mute to prevent any background noise. After the speaker's remarks, all lines will be opened in order for you to ask questions. If you should need assistance during the call, please press star 0 on your telephone keypad, and an operator will come back to assist you. Thank you. Amanda Hopping-Wynn, you may begin your conference.
Thank you, Sarah. Hello, everyone. I'm Amanda Hopping-Wynn, Associate Director here at the National Abandoned Infants Assistance Resource Center located at the University of California at Berkeley. Funded by Health and Human Services Children's Bureau, our mission here at AIA is to enhance the quality of social and health services delivered to children and families affected by substance abuse and or HIV by providing quality training, information, and resources to the professionals who work directly with these families. Today's call, The Long-Term Effects of Prenatal Substance Exposure on Children, will be presented by Dr. Barry Lester. Dr. Lester is a professor of psychiatry and human behavior, professor of pediatrics, and the founding director of the Center for the Study of Children at Risk, Brown University, Alpert Medical School, and Women and Infants Hospital. The focus of Dr. Lester's research is on mechanisms and processes that determine developmental outcome in children at risk due to biological and or social factors. He has studied the effects of prematurity, growth restriction, malnutrition, prenatal substance exposure, and maternal psychotropic medication during pregnancy using longitudinal multi-site and cross-cultural designs. His work has shown that biological factors do leave their footprint on later development and that environmental factors can exaggerate or lessen the impact of biological insults. Dr. Lester's current work includes study of the molecular basis of behavioral development through epigenetic changes that result from prenatal insults and environmental adversity. At the Center for the Study of Children at Risk, many of these research findings are translated into assessment and treatment programs. Dr. Lester has served on NIH study sections, the NIH National Advisory Council on Drug Abuse, the NIH Director's Pioneer Award Program, and the College of the Center for Scientific Review at NIH. He is past president of the International Association for Infant Mental Health and the author of more than 200 scientific publications and 16 edited volumes. We're very fortunate to have him here with us today. During the webinar, Dr. Lester, is going to pause at a few points for questions. At that time, we will open all lines, and you'll be able to ask him directly. As an alternative, if you'd like, you can chat your questions to me, Amanda Hopping-Wynn. In that bottom right-hand corner, you'll see a chat box, and you just find my name in the Send To box. Send me your questions, and I can address Dr. Lester if you prefer that method. So now I turn the call over to Dr. Lester.
Oh, hello, everybody, and thank you very much for inviting me. Actually, this is Dr. Brown. It's great that you're out there in Berkeley. I'm really, really jealous because in Providence, in Rhode Island, we still have lots of snow. And, I could use some nice weather.
Not to rub it in, but it is about 65 and sunny here.
Oh, God. I forgot what the sun looks like. Is it that little yellow thing that sometimes is up there? All right. So let me get started. Basically, this is a summary of what I'm going to be trying to get across, which is the idea that, there have been concerns for a long time about are there prenatal exposure effects and how big are they and how do they interact with environmental factors. And there's been 20, 30 years of research looking at this and with varying kinds of outcomes. But, basically, the big answer is yep, there are drug effects. They're not as large or as devastating as, people initially thought, especially when we're thinking about things like cocaine and methamphetamine. It's important to realize that, not all drug-exposed children are affected, so that when you see studies and, it says, this group has, more cognitive problems in that group, you're only talking about an average of, a sample, and that doesn't mean, every single child is affected. Some are, some aren't. Um, you'll also see that some of the effects actually don't show up until kids get to be older, and we call those latent effects, and probably has to do with the fact that some of these drugs are affecting areas of the brain that are really not activated in infancy or in infancy and early childhood. Areas of the brain that control things like executive function and planning and activity that really don't come online until childhood. So in some cases, you actually see effects getting stronger as children get older. And, the last thing is that drug effects can be treated. I think we've known this for a long time, but it's been hard to, I think, convince policymakers. Kids with prenatal exposure are definitely at risk. Children can be at risk because of different factors, in this case biological, and so certainly prenatal cocaine exposure would be an example of a biological effect. And children can also be at risk for social reasons. And, kids who grow up with prenatal exposure often grow up in environments that are not particularly optimal. So you have the combination of, biological and social factors that are really determining the outcome of the child. So just in general, all drugs of abuse, it doesn't matter what they are, whether it's, alcohol, tobacco, or illegal drugs, cocaine, methamphetamine, each drug does have its own separate impact on the brain, but they also share one common impact, which is that they affect the what's called the brain reward pathways, the dopamine pathways, and these brain circuits are the circuits that light up or that are activated when you feel good. So they get activated, with food, with music, with sex, and anything else that gives you pleasure, and also, particularly with cocaine, one of the things that cocaine does is that it creates even more so the release of a neurotransmitter called dopamine, which is the one of the transmitters that facilitates communication between neurons. And dopamine can be a good thing or it can be a not so good thing. And basically what happens with drugs like stimulant drugs in particular, like methamphetamine and cocaine, you have two big effects, two things going on at the same time. One is what's called vasoconstrictive, which means that these drugs constrict the arteries in the uterus, and what that does is it reduces blood supply to the fetus. If you reduce blood supply to the fetus, you're reducing nutrition, and you're also reducing oxygen so the baby can be oxygen deprived or hypoxia. So that's one thing that these stimulant drugs do. The other is what's called the neurochemical effect, and this is back to the dopamine issue. Normally, what happens is the dopamine gets released, that's these little red dots here, and then it does its communication, and then it sort of gets sucked back up into the cell, because you don't want to have too much circulating dopamine in your system. Too much can make you do risky things, and it's associated with a lot of different kind of high risk behaviors. But what methamphetamine and cocaine do is they block this reuptake. They prevent the dopamine from going back into the cell, so the brain gets flooded with dopamine and it just stays there. So those are really important major effects, and most of the research on drugs like methamphetamine and cocaine are based on that kind of understanding. Later on in this talk, I'll tell you about somewhat of a different way to think about these drugs, and in a way that has more to do with stress and impact on the health of the child. So I'm going to give you a summary of what we know about methamphetamine, and then we'll talk about cocaine, and then we'll talk about some other ways of thinking about these drugs. Methamphetamine, depending on where you people on this call are sitting, there are over 250 million users in the world. It's the fastest growing rate of growth of illegal drug, and it's also in the United States. It's very interesting because you almost never see it east of the Mississippi. So if you're east of the Mississippi, Hawaii, that's where the most of the methamphetamine use is. Here on the East Coast, we never hear about it, and it's amazing to me because I'll be at meetings or talk to a colleague or something, and talk about some of the methamphetamine research, and they'll say, "What? Methamphetamine? Is that a diet drug or something?" I mean, it's almost unheard of. I mean, it's we're seeing more and more of it, little bit at a time. In our hospital, which delivers almost 9,000 babies a year, I've never seen a methamphetamine exposed baby. So it's just not common around here. Okay, so in terms of what we know about the effects of prenatal methamphetamine exposure, unlike cocaine, where there were lots and lots and lots of studies that were started, and we really have a huge amount of information about prenatal cocaine exposure, we do not have that for prenatal methamphetamine exposure. There was one old study done years ago, but it was, very poorly done, kind of a retrospective thing. So this study here, called IDEAL, the Infant Development, Environment, and Lifestyle Study, is actually the only longitudinal prenatal study of methamphetamine. And this study was started primarily because a lot of the people, a lot of the scientists in the West were seeing all these longitudinal studies of cocaine and, writing to the NIH and say, "Hey, guys, you don't know what's going on out here. We don't see cocaine. We're seeing methamphetamine." So the NIH decided, that it was time to do one of these longitudinal studies. So we are studying prenatal methamphetamine exposure in 4 sites: Iowa, Oklahoma, California, and Hawaii. And then after we got started, we added Auckland, New Zealand as a 5th site. You may ask, why Auckland, New Zealand? If you've seen Lord of the Rings, you know why you have to go to New Zealand. There are also scientific reasons. The Kiwi, as they call themselves, pride themselves on being really, really excellent chemists. So they make the purest methamphetamine that you'll find anywhere. So that was one reason we wanted to go there. second was that socially they don't have the distribution of social class and poverty that we have here in the States, and everybody has access to the same healthcare system regardless of your income. So that huge confounder of social class that you see in the United States, you don't see in New Zealand. And the third thing is that in the U.S., it's fairly common, depending on where you are, to have babies removed from their bio moms and put into foster care because of prenatal drug exposure. In New Zealand, that is a cultural taboo. Never, ever, ever happens. In fact, I was once giving a talk down there, and telling them about these issues with child removal, and somebody stood up in the audience and said, "If anybody in this country ever tried to take a child away from its mom because of prenatal meth use, there'd be a national riot." So again, that's another one of those confounds that we can eliminate by going to New Zealand. This is just a summary of some of the research that's come out of the study. We have about almost 500 kids enrolled in the study, half methamphetamine exposed and half not. And as I said, they're in those 5 sites. But early in the early papers, we basically found some newborn effects where the methamphetamine babies were showing poorer sucking. They spent more time in the neonatal intensive care unit. Their length was a little shorter, and they weren't premature. These babies do not tend to be preemies, but there's a higher incidence of small for gestational age, which means they're term, but they're underweight. So that's usually because of some kind of nutritional deprivation in the 3rd trimester. So they're term babies. They're just small for being term. Then we have a neurobehavioral exam that we actually developed for the NIH, and on that exam, we found that there were more stress signs in the methamphetamine kids. They were lower arousal and more lethargic. They had a poor quality of movement, and then their motor control the defined motor control was worse between ages 1 and 3. Now, when you see here low arousal, more lethargy, sometimes you think that contradicts the sort of stereotype of the screaming, back arching, drug exposed baby. What's interesting about the methamphetamine exposed babies is when you first see them in the nursery, and if they're sleeping, they are in such a deep sleep. I mean, they're hard to arouse. I mean, they really look like they're deep, very deep down under, and they're very, very hard to arouse. But once you do get them up, they scream. Okay, and so what you don't see is a lot of the sort of quiet, alert behaviors that come in between sleeping and crying. They seem to be like light switch babies. They're either on or they're off. And then we saw some decrement in length between ages 1 and 3. Just wanted to fast forward, to 3 and 5 years of age in terms of what we're seeing in these kids. We looked at behavior problems on something called the Child Behavior Checklist, where you can look at, externalizing, acting out kinds of behaviors, including things like ADHD, internalizing depression, and this is something that moms fill out, and it's used clinically, so it's a good instrument. And what you see here is that the methamphetamine kids showed more externalizing behavior, significantly more externalizing behavior at 5 years of age, and they also showed more emotional reactivity and anxious depression at both 3 and 3 years of age and 5 years of age. And there's a subscale on externalizing of ADHD and again, we saw more ADHD problems in the methamphetamine exposed group. So one of the things that we're interested in looking at, and when I mentioned before that some of the effects don't show up until later, there's this concept of executive function, which has to do with, planning and attention and impulsivity, which is obviously related to ADHD. It's not particularly correlated with IQ. I mean, IQ is something different. Executive function has more to do with, how you were able to plan and negotiate in the world, so to speak. So here's a test. It's called a, CPT, continuous performance task. So the kid is sitting in front of a computer screen, and they're going to see pictures of one of these things, a horse, a house, scissors, boat, bicycle, whatever. And what they're told is every time you see one of these, hit the button, right? Hit the button when you see one of these. But if you see this, if you see a soccer ball, don't hit the button, okay? So this is called a go, no-go task. And what it measures is, impulsivity and inattention, because, these things are flying by, the kids are hitting the button, this thing comes up, they're supposed to not hit the button, but they can't help it, they hit the button, and so they make an error. So that tells you something about impulsivity. And on this particular test, you do get a measure of impulsivity, an ADHD index, and you can see here that the exposed kids, have a much higher index of ADHD than the unexposed kids. Having said that, I mentioned earlier that not all of these kids show the effects, and so you can see that you've got 25% here and 15% here. So one of the things that has become very, very interesting about drug-exposed kids, but other kinds of kids with behavior problems as well, is the fact that the problems that you see in these kids, so things like impulsivity, aggression, sensation-seeking, irritability, ADHD, all these different kinds of behavior problems, we tend to think of them as independent things. Oh, the kid has ADHD. Oh, the kid has this. Oh, the kid has that. But in fact, they're highly related. I mean, a lot of these behaviors tend to, kind of run together. They're highly correlated. So that's led to the development of this construct called neurobehavioral disinhibition, which is something that's becoming more and more used in psychiatry. And basically the argument is that these behaviors, what they do is they represent this underlying deficit in behavior control. I mean, there are different aspects of it, but in general, you're looking at an underlying deficit in behavior control or poor inhibitory control, and it's really almost a single dimension, but it shows up as these kinds of behaviors. And then, so when you look in the literature and people measure this neurobehavioral disinhibition, it's related to substance use in adolescence, early age of onset, and substance use in adulthood. So it's turning out to be a very useful construct, and we think it's very useful in terms of understanding the effects of prenatal drug exposure. Here's one of those slides that you look at and you go, oh my God, what, are you kidding me? You don't have to look at all this in detail. It's to show you that if you look at that, neurobehavioral disinhibition has essentially two components to it. One is behavioral and emotional control. Again, things like poor inhibition, aggression, et cetera, et cetera. And the other is what I mentioned before as deficits in executive function. Okay, and these are just different ways of measuring executive function. But the disinhibition construct has both a cognitive piece to it and a behavioral piece to it. And what this is showing from our research is that there's a relationship that prenatal exposure predicts, at 5 years of age, it predicts behavioral and emotional control, which in turn predicts the cognitive function. And there's no independent prediction of methamphetamine on executive function. So the pathway, and that's what the interesting part about it is, the pathway is through the poor behavior control, with methamphetamine, resulting in poor behavior control, which is then related to poor executive function. So it kind of tells you developmentally what the pathway and the onset of this is. So another big player in this, I mentioned before, is the environment. A lot of these children grow up in real environmental adversity. So, we look at things like, depression, low SES, substance use of the caregiver at home, poverty, quality of the home, psychological problems. And what we're interested in now is, okay, so we know that there are effects of prenatal methamphetamine exposure on this construct of neurobehavioral disinhibition, right? More exposure results in more disinhibition or more problematic behavior. But if you then introduce a measure of environmental adversity, so what we've done here is just taken created an index of adversity based on these particular factors. What you find out is that there's still a significant independent effect of methamphetamine, but there's also an indirect pathway through environmental adversity. So methamphetamine is related to adversity. Adversity is in turn related to disinhibition. Now you can see the effect here is smaller than the effect is here, because this drops because we've added in the adversity index. But the important point is that disinhibition is related to both methamphetamine itself, but also augmented or exacerbated when you add environmental adversity. So I'm gonna move now to looking at cocaine. This might be a time we could stop for a few questions before we, switch from methamphetamine to cocaine. People have their different favorite drugs. So.
Sounds good. Sarah, can we unmute lines for questions?
All lines are now open.
Does anybody have any questions for Dr. Lester at this point?
This is Tangi. Can you hear me?
Yes.
Okay. I wanted to find out when he was talking about in the last slide about the the slide is gone now. But anyway, I wanted to find out if the pattern of the caregiver can affect the diagnosis of ADHD or ADD.
The pattern meaning the attachment?
Not the attachment, but the pattern of the caregiver, meaning partly low attachment, but also the interactions between the parent and the child, or their lack of, and also the structure, meaning there is not a lot of consistency in the routine, and could that confuse or impact whether or not a child could be more than likely to be diagnosed with ADHD?
Well, the short answer is yes. I mean, we look at a number of parenting factors, and so inadequate parenting is certainly part of the adversity package. It may have to do with maternal mental health. It may have to do with parenting itself or attachment. So it is one of the major players. In this particular case, it's not pulled out on its own, but certainly in other studies, it's been shown to have an effect. Having said that, it's in the context of the prenatal drug exposure. So what we're talking about is the combination. It's not just the parenting in itself; it's the combination of the parenting and the prenatal drug exposure that's going to result in the ADHD.
Okay. Thank you.
Sure.
You have a question, please. Hello.
Hello.
Hi.
My question is, have you studied the effects of early intervention on meth-addicted babies? And so did the change in the cognitive abilities and those things that you saw when they were 5 and up, did you see a change in those studies with
A change with early intervention?
Yes. From that 5-year-old, if there was early intervention during that time, did you see any changes in their cognitive ability and things like that?
Well, this is not an intervention study, so we're not we're following these kids developmentally, to try and understand their developmental course. So we're not doing interventions with these kids. We do keep track of the kinds of services that they get, but they don't all get terrific services.
I have a question.
Yeah. Okay.
Okay. How did you define adversity?
Are these people looking at the slides?
Okay, so I just put back the slide with the
Thank
adversity index.
All right.
So it's looking at psychological problems in the caregiver, depression, low social class, whether or not the caregiver is using substances at home, the quality of the home, and poverty. And the reason the word caregiver is here is because not all of these kids are with their biological moms. And one of the big factors that plays a role in the outcome of these kids, not in this particular study, in this analysis, is just the number of placements. The number of caregiver changes. So that's one of those environmental factors. So I think maybe we'll
I have
move
one
on.
quick question.
Okay.
I'm just wondering, and I was a couple minutes late, so I may have missed this, but did you talk about any effects? Were there any in infancy? I work in a birth center, so I'm just wondering, as far as working with parents and foster parents at discharge from the hospital, is there anything that they should be looking for or watching out for with these infants? I know, being lethargic, is poor feeding, is that anything during infancy?
Yeah, I did show some of the initial newborn effects. They tend to be small for gestational age, but full term but underweight. On our neurobehavioral exam, they seem to be more stressed, and I think you used the word lethargic or under-aroused. We see that also. They have trouble with attention, but they're not. One of the things that always comes up about these kids is how different or how remarkable are they in the sense that can you walk into a nursery and be able to say, oh, I can tell that kid's prenatal methamphetamine, or it's not that you can necessarily pick them out, from other babies in the nursery. The kinds of effects they show are, not really dramatic, and I said earlier on, some of them actually get stronger as they get older for other kinds of reasons. But yeah, there are definitely newborn effects.
Okay, thank you.
Okay.
Hey, Dr. Lester, this is Harriet Gaines, Centerpoint Residential Women and Children's Program, and I just want to say thank you for this presentation. This data is consistent with the observations we've made of the children served at our program, children between the age of 5 who were predisposed in vitro to substance use. And this is great, and it just makes me feel reassured that the interventions that we are applying are really effective. So thank you so much.
Great. Thank you.
And Sarah, with
Okay.
that,
Yep.
can we mute the lines again, please?
All lines have now been muted.
Thank you.
Okay.
Back to you, Dr. Lester.
So we'll switch to cocaine now, and these headlines are from the mid 1980s when everyone was so afraid of the long-term effects of prenatal cocaine exposure. So these are just some of the headlines that were around then, and we all know now that a lot of this was exaggerated, and a lot of these mothers were prosecuted unnecessarily, and it was I think it was a not good time in our history in terms of social responses to this kind of problem. But what it did do, the silver lining, I suppose, what it did do was it triggered an enormous amount of research. I mean, there were 10 or 15, major. I mean, unlike with the methamphetamine, where the only one we have is our ideal study, there were at least 10 or 15, major longitudinal studies that were started because of the tremendous concern, about the outcome of these babies. So here I can show you, because we have so much information now, I've broken down the different effects that you see across studies. So some studies show, effects on behavior problems, IQ, executive function, language, et cetera, et cetera, et cetera. And the green, so here you're looking at the number of studies. So you can see there were 25 studies that looked at behavior problems. And the green shows you how many of those studies showed statistically significant cocaine effects. Okay? So this tells you a couple things. It tells you, well, what the most studied things have been, behavior problems, IQ, executive function, language, and stuff like, somebody mentioned relationships before. Very little. Substance use is just starting. Very little on psychopathology. So that's one thing you can see. Second thing you can see is, whereas half of the studies found significant effects in behavior, only about a third, so more studies that found non-significant effects for IQ than significant effects. But look at executive function, finds more, proportionally more effects than any other domain, language also. And then there are a couple studies on school function. And, the executive function deficits have to do with, again, I circle back to what I mentioned before, that one of the things that we think that's going on with these drugs is that they affect these areas of the brain, the prefrontal cortex and the cingulate, et cetera, that are not online or not activated in infancy. So you may see relatively small or no effects in infancy, but then these effects are really latent. They sit there, and then when these other areas of the brain come online, that's when you start to see more of the drug effects. Of course, that's also when the environmental effects are going to be stronger too. So, as part of all of these longitudinal studies, the NIH decided that, this is such a complicated problem, and usually when you're doing these studies, you do, let's call it an investigator-initiated study, right? Somebody sends in a grant, and, if they get funded, they do the work. But sometimes the NIH decides that there's a problem out there that is just way, way too big for one person to do, and it needs to be done, by the NIH itself. The breast cancer study was one of those, and that's what they did with cocaine. And so they created this study called Maternal Lifestyle Study, or MLS, and it was conducted in several different sites. I'm the PI of the study, but I'm sort of a hired gun. I'm sort of doing this as with their funding, and so we started out with almost 1,400 kids, 658 cocaine exposed, 730 were not exposed. We matched them pretty carefully on medical and demographic factors. This was one of the first studies that used meconium toxicology to detect drugs. Meconium is the first stool that the baby passes. It's that black sticky tarry stuff, and what it is all of the residue that's been sitting in the gut since 20 weeks gestation, and you can get drug toxicology out of it. So the beauty of it is you don't have to rely on self-report. Any drug use that went on during that time will show up in the toxicology assay, and it's particularly useful for women who use drugs but say no. So in this particular study, there were 33% of the women denied using cocaine, but they were meconium tox positive. The other thing we did here was, you always hear, and it's very, very true, that, women who use cocaine during pregnancy don't just use cocaine. They're polydrug users, so it's really hard to know whether you're looking at the effect of all these drugs together or other drugs. And so what we said was, our control group, instead of having a control group with no drugs, our control group has alcohol, marijuana, and tobacco, but no cocaine, right? So one group has cocaine plus alcohol, marijuana, tobacco. The other group has alcohol, marijuana, tobacco only. So if we see differences, we assume that it's due to the cocaine. Just demographics, nothing that you don't know. They're similar, but we're looking mostly. Cocaine you see more in black inner city cities. So 76% were black, non-Hispanic. 79%, 48 of mothers were over 26. Most of them single, on Medicaid, less than a high school year of education, and we had 11 or 12% babies born less than 32 weeks gestational age. So I mentioned that neurobehavioral disinhibition thing before, and again, it's something that seems to be very, very useful in this cocaine cohort as well. So when the kids were between 11 and 13, that we administered a whole bunch of tests. Here again you see the child behavior checklist. The DISC is a psychiatric exam that gives you diagnosis. So we have ADHD, ODD, CD, depression, and then these are acts of delinquency that the child admits to. It's called things you have done, and the kids endorse whether or not they've done different kinds of delinquent acts. And then, we have two cognitive measures, two measures of executive function, because I mentioned before that this disinhibition construct has both these behavior, externalizing kinds of behaviors as well as poor cognitive control. So if you look at, so you compute, mathematically, you compute this disinhibition factor, okay? So the factor itself is composed of all of these measures put together. So every kid has one score, a disinhibition score. The higher the score, the more disinhibition, or the worse off you are, so to speak. And you can see here that if you're cocaine exposed, your disinhibition score is much higher. It's very positive. If you're not cocaine exposed, your disinhibition score is much lower. So again, these behaviors, tend to travel together, to be highly related, and to be related to prenatal cocaine exposure. So here we're looking at when these kids themselves start using substances. Okay, so at what age? What's the age of first use or age of onset in this case of these cocaine-exposed kids? And here we're looking at up to 16 years of age. Okay, and this survival, called the survival probability chart. It's sort of counterintuitive, because what it's telling you is that as you go down, you're looking at more use. Okay, so up here there's 100% of non-use. Here there's 85% of non-use, or 15% use. But the important thing about this is you can see, if you follow the steps down here, you can see the age of first use of the two different groups and where they tend to split, if there's a difference between the cocaine exposed and the unexposed. And the steps start to separate around 12 years of age, where you can see that that more of the cocaine-exposed kids are starting to use, in this case, tobacco. So this is showing that prenatal exposure, 16 years later, is predicting kids who are going to start using, in this case, tobacco, that the age of first use is earlier. And age of first use is a good predictor of later substance abuse disorder. And as you know, at this age, the drugs that kids are using are typically alcohol, tobacco, marijuana. I mean, you don't see, hard drug use at this age very much at all. The second thing that's interesting about this is, like I said, this is the statistics. So it's saying if you're cocaine exposed, you're almost 1.5 times more likely to start using drugs earlier. So that's just saying, what does cocaine do? But then, if you put the disinhibition in, right? So cocaine, in your model, in your statistic, whatever, you've already shown that cocaine affects the age of first use. If you then introduce the disinhibition measure, it knocks out the cocaine. Okay? Cocaine is no longer effective, significant, when you put in disinhibition. So what this means is it's not that cocaine is affecting age of onset directly. It's that cocaine is affecting disinhibition, and it's the disinhibition that is actually taking over and affecting when these kids start using tobacco. And then the other thing is, whenever you do these kinds of analysis, you look at environmental factors also, right? Someone mentioned before maternal psychopathology or poor parenting. So you look at all those things. And in this case, there was a separate effect for child abuse. In fact, the child abuse effect is larger than the disinhibition. It's twice as large as the disinhibition effect. So, if you were abused as a child, your almost 2.5 times more likely to start using tobacco earlier, and that's independent of the disinhibition effect. So both of these things are, in a sense, going on at the same time. You can do the same thing for marijuana. Here it's even a little more dramatic. You can see the separation in the steps. And again, it's around 12, where first it looks like the cocaine-exposed kids are, 1.37 times more likely to use earlier. You then introduce the disinhibition. It knocks out the cocaine. Cocaine is no longer significant. And then you get two more additional effects. Boys, right? So boys are more likely to start using earlier, and kids who are exposed to community violence. So you can see in each of these, different environmental factors come into play that exert their own influence in the context of the cocaine. So it's a reflection of both the environmental adversity in which these kids are growing up, as well as the kind of interaction between drugs and environmental problems. This is illicit drugs. I mean, illicit is anything but cocaine and so again, it's the same story. Earlier onset with cocaine, you introduce disinhibition, you lose the cocaine effect, and once again, you get an effect for boys, and you get an effect for community violence. The other thing we started to look at 16 is sexual issues. So if you're looking at sexual contact, any sexual contact by 16, again, I hate to sound like a broken record, but it really is intriguing how the same story comes up. Okay, so you get first your cocaine effect with more sexual contact in the 2 green here in the cocaine. Put in disinhibition, it knocks out cocaine. Boys come in, so you can see there's more sexual contact with boys. But once again, if you're exposed to community violence, there's an effect. And if the caregiver is using tobacco at home, there's also an effect. So this is the first time we've seen this. And, because tobacco is smoke, this could be due to secondhand smoke. It could also be due to certain parenting characteristics about, people who smoke a lot of cigarettes that we don't know about. But it certainly comes in there. And then if you looked at unprotected sexual intercourse, so basically no condom use, again, you first get a cocaine effect, knocked out by disinhibition, higher in boys related to community violence, and the postnatal tobacco use. So, we're seeing the same kinds of effects for, early risk sexual behavior as we are for use of drugs.
Before you move on real fast, can I just ask a clarifying question about community violence? We have some participants who are wondering if you could just define that term community violence. Is that related to gang activity only, or is it broader than that?
It's a questionnaire that is administered to the kids, and it has different kinds of categories of community violence. It could be gang related. It could be seeing people fighting. It could be things like guns, and I mean it's a checklist of a lot of different things that you look at, and you add it up and you get a total score.
Okay, thank you for clarifying.
Sure. So, this kind of, I love to be simplistic, right? I mean, this stuff kind of boils down to the fact that you're looking at how the environment, amplifies drug effects. So you have the combination of the fetus being exposed to the drug, having those, let's say, neurochemical and vasoconstrictive effects affecting the brain, and also things like, violence affecting the kid being exposed to violence, and it creates some kind of a perfect storm, which has been referred to as toxic stress, a toxic environment, which the thought being that these kinds of toxic environmental exposures can have long-lasting effects on different physiological systems and maybe predispose kids to health disorders as well as adult psychopathology. Let's see. This is probably another good break point.
Sarah, can we unmute the lines?
All lines are now unmuted.
I have a question for you. Can you hear me?
Yep.
Yes.
Great. Back at the beginning when you were talking about the MLS study design, you mentioned that the group had cocaine and or opiate use. And then that's the last thing we hear about opiate use. We're in Kentucky, and we have a whole lot more opiate use than cocaine use for most of the state. And I'm just wondering if you could say that your outcomes would also apply to opiate exposure without cocaine.
Yeah, the opiate stuff that you're seeing today, I would imagine mostly what you're talking about is abuse of prescription meds, OxyContin and that kind of thing.
Actually, we have a lot more heroin these days, so.
Oh, okay. So because this data was collected in the early 1990s, and the reason that we said cocaine and or opiate was because there was so little known about the epidemiology of cocaine at the time. We thought that it would be hard to find cocaine users who didn't use opiates, right? It turned out that wasn't true at all. I mean, it turned out that, we didn't need the opiates at all. There were actually relatively few of them. And so we don't have very many in this group. We've done some analysis where we've separated the cocaine only from the cocaine opiate. In fact, our first paper on newborn neurobehavior did that. And yes, in that paper we did find that the combination was worse than the cocaine alone. As these kids have grown up, because our focus was our job, so to speak, our mandate from the NIH was to really figure out this cocaine stuff. What we would do was either to drop the opiate kids from the analysis, so that the follow-up data that I showed you did not have the opiates in there. There were about 110 of them, I think, or use it as a covariate. So in either case, it was taken out, and it has not been examined separately.
Would you do that, please?
Yeah. Hang on, I'll be right back, and
we get a lot of questions in Kentucky from people about, well, what are the long-term effects of opiates, and there's so little data about that would be so valuable if somebody could look at that at some time.
Yeah. No, I know the literature. I mean, there are probably 3 or 4 real longitudinal studies of heroin opiates, and they're small samples, and there's just very, very little that's been done about that. So, I'll send you the data. You can look at it.
Great. Thanks.
Right.
Also, we had some requests for references for these studies. If you could send them to me afterwards, Dr. Lester, I can make sure they're available to participants.
Sure.
Thank you.
Okay.
We do have about 30 minutes left for this presentation, so I leave it up to you, Dr. Lester, if you want to continue taking questions or if you'd like to move on.
Yeah, let's move on. I'll try and leave some time at the end.
Okay.
Okay, so here's the slide I showed you earlier on about the different kinds of major mechanisms of methamphetamine, cocaine, the neurochemical and the vasoconstrictive. There's a different, sort of a new way that people are starting to think about these drugs, and there's a lot in the literature these days about prenatal stress, and there can be all kinds of stressors. I mean, depression can be a stress. Poor nutrition can be a stress, and how prenatal stress can have outcomes on particularly the HPA axis or neuroendocrine system that determines how you react to events in life, and again has a lot to do with psychopathology. So you can think of drugs in general not only in terms of their, again, vasoconstrictive and neurochemical effects, but also in terms of how they might be a stressor in utero, and how stress alters something called fetal programming. And fetal programming has to do with this idea that's it's a very, very hot topic right now, and basically has to do with the idea that the fetus is capable of reading cues from the internal environment. Let's say reading cues, from the intrauterine environment, and reprogramming itself, or kind of anticipating what kind of environment it's going to be born into, and adjusting its physiological systems accordingly. Okay. Now that may seem totally, fantastical and impossible, but there's a fair amount of data that this actually happens. And so one of the things that people are looking into is how this concept of fetal programming is related to something like an insult of drug exposure. And again, what are the potential mechanisms and what are the long-term effects? So there are certain genes in the placenta, and there are two of them that are of particular interest here. One is called 11 beta, and the other is GR, which is a glucocorticoid receptor. And what these genes can do is, by affecting how much cortisol the fetus is exposed to, disrupt the HPA axis and result in this kind of behavioral dysregulation. So basically, I mean, I just, not going to go into a lot of detail here, but what happens is you have, adversity in the intrauterine environment. Can be drugs, can be toxicants, can be smoking, can be stressors. Again, depression, maternal, psychopathology. So this is putting drugs in an entirely different context. It's putting it in the context of life stress in a much broader context, very relevant to the kinds of adversity in which a lot of these babies are growing up. So the fetus is exposed to a certain amount of cortisol, and cortisol is, it's an important thing. It's something that gets the physiological system moving when it has to. But exposure to too much cortisol is dangerous. In fact, it can be toxic. So these genes, 11 beta and GR, what they're responsible for is they take the cortisol that the baby is baby's being exposed to, and they break it down into cortisone so that it's inert, right? It's inactive. Okay, so they basically lower the amount of cortisol that the fetus is exposed to, so it doesn't get to dangerous levels. Okay, if that doesn't happen, then you get a disturbance in this HPA system, and so one of the thoughts is that these things like drugs and other stressors, what they do is they prevent these genes from breaking down the cortisol. They prevent these genes from doing their job. So the danger is that the fetus then becomes exposed to too much cortisol, and they're dysregulated right at birth. So this whole thing comes from this field called fetal origins, and the idea of fetal origins is that the susceptibilities. It really started out in terms of looking at adult disease, but that the susceptibility to certain kinds of diseases is programmed in utero initially as due to fetal undernutrition. And so the story is, it's a very interesting story and very well replicated. In 1944 and 45, the Nazis starved the Dutch people, and so they underwent this winter where they were in starvation, and so obviously a lot of the fetuses were malnourished. And then someone named Barker came along and started doing follow-up studies, and he looked at these people who were now in their 30s and 40s and 50s. I mean, this is still going on and on and on. And what he found was that the fetuses, that the babies that were born underweight, okay, and some you can either look here or look here, but the babies who were born underweight are more likely to have diabetes when they're in their 30s and 40s, or in this case, you're looking at the combination of if they're low birth weight and heavy, these are women, they're more likely to have metabolic syndrome. And the question became, well, why? How does that happen? And the way it happens is this idea of fetal programming. Okay, so again, this is not fantasy. This is real. But the idea about the explanation for why these metabolic syndromes happened was that the fetus is being starved in the famine, right? The fetus is being starved, so it's saying, uh-oh, I'm going to be born into a nutritionally deprived environment. There's no food here. There ain't going to be any food out there. So I need to prepare myself physiologically. I need to slow down my metabolism. I need to alter my metabolic system to be able to be prepared to survive in a nutritionally poor environment. But what happened? What happened was the famine ended, and these babies were in fact born into a nutritionally adequate environment. But they were programmed now for the wrong environment. Okay? And so because the programming was more or less fixed, they wound up later on having these different kinds of diseases and disorders, because there was a mismatch between their programming, how they programmed themselves, and the postnatal environment. So the original idea was, you had this reduced fetal growth. It changes the structure and function of the physiological systems due to this fetal programming, and that can increase the risk for adult disease. So this whole idea is the idea that we have this thing called developmental plasticity, which is part of what makes humans special, right? That we have this ability to change structure, to change the actual structure of our bodies as well as function in response to different kinds of cues that we get in the prenatal environment. And that's evolutionarily very adaptive because it means that you can have a wide range of phenotypes of different kinds of behaviors in people and individual differences that are going to develop from a single genotype depending on environmental factors. And the fetus is now making these adaptations through programming to prepare for the postnatal environment in response to the kinds of signals that it's reading. So all of this was happening 20 years ago, 15 years ago, in mostly in medicine. And then people started thinking, oh, that's interesting. If this fetal origin stuff is useful for health and medical outcomes, would it also relate to behavior? I mean, is there any evidence that this fetal origins theory could explain the etiology of, let's say, mental illness or substance abuse, et cetera, et cetera? So it turns out there is some data in the literature showing that low birth weight, in fact, is related to schizophrenia and depression and to psychological distress. So that sort of fits the Barker, the babies who are undernourished. But then the real, to me, the most exciting part of this is to say, okay, can these effects or can similar effects, we know that it works with undernutrition, but could it also work with other environmental insults like drugs, like prenatal cocaine or methamphetamine exposure? And if so, what might be the underlying mechanisms? And the main mechanisms that people are studying today is what's called epigenetics. And epigenetics relates to the sort of molecular basis for these kinds of changes. And I promise I'm not going to spend a lot of time on this, but it's really fascinating. So epigenetics is the idea that you can create changes in gene activity or what's called gene expression without actually affecting the structure of the gene itself, the gene sequence. So basically, the gene stays the same. It's exactly the same, but it doesn't do what it was programmed to do. Okay? Genes can be silenced by preventing gene expressions. Genes can be enhanced by increasing gene expressions. And some of these changes can be actually transmitted to the next generation, and some of them can be reversed. Okay? And this is not science fiction. This is real, and it's happening, and there's a huge, this is probably the one of the hottest area in sciences today. And how it works is through different kinds of mechanisms, but the one that is studied more than any other is called DNA methylation, which you can think of as gum on a light switch. Okay, if something is methylated, it's like turning off the light switch. It shuts down the gene, and methylation is a normal developmental process. It's already been implicated in cancer. There are already epigenetic therapies for cancer where they induce epigenetic changes to combat cancer growth. Okay, and again, this is the last of these, but basically, what methylation means is that you've got your gene sequences here, the different amino acids, and the DNA will attract this what's called a methyl group, and the methyl group puts a tag on top of the gene. So the gene is the DNA is unraveling so it can copy itself. So all this stuff happens during cell division, and this methyl group gets stuck on top of the gene, and it shuts it down, or it reduces the gene expression. And it's a well-known and very well-studied mechanism, and people are literally just starting to apply this kind of epigenetics to behavior. I mean, like I said, there's a lot of it with medical issues. There's also a fair amount with rodent animal studies, but the application to human behavior has literally just started. In 2011, we did a conference for the New York Academy of Sciences on behavioral epigenetics. It was the first ever. I mean, it was the first one of its kind. So that just shows you how new this stuff is. So the idea of relating epigenetic changes in utero to actual behavior is what we're working on and other people are working on. And here's just an example. This is one of those genes in the placenta, the 11 beta that breaks down the cortisol. So what this is showing is that if you have a lot of methylation of this gene, meaning you're preventing this gene from doing what it's supposed to do, which is break down the cortisol, when this baby is born and you test this baby on the neurobehavioral exam, they have a much poorer quality of movement. Their motor behavior is poor because they've been exposed to too much cortisol due to methylation of 11 beta. And then here's the other gene I mentioned. GR is the glucocorticoid receptor. It does something very similar. The cortisol, binds to this gene so that if this gene is methylated, cortisol doesn't bind. You get more cortisol floating around, and now you see the baby's born. And if there's a lot of methylation, you've got many more of these babies who have very poor attention scores if there's high methylation, or good attention scores if there's low methylation. So again, shutting down this gene through methylation exposes the baby to too much cortisol, which will then result in compromised neurobehavior. So what that sort of gets us to, again, is taking the prenatal exposure literature and putting it in this larger context of environmental adversity and different kinds of stress, where the model would say something like, you have the neuroendocrine system, the HPA axis, which is going to be affected both by something like drugs and the environment, which is going to result in some kind of neurobehavioral disinhibition and potentially substance use in adulthood. And the idea is this concept of toxic stress that, with over time, with prolonged, if you keep on banging away at this system, if you keep on stressing the neuroendocrine system, which is supposed to respond to emergencies and fight or flight reaction, you're eventually going to wear it down, and you're going to affect the overall stress response system, and that leads to some of these adverse behaviors. So there's actually some evidence for this going back to the maternal lifestyle study. So here we took kids who, this is 11 years of age, and we put them through a stress paradigm, and you collect saliva before and after. You measure cortisol and saliva, and what happens is the cocaine-exposed kids show a decrease. I mean, normally, saliva or cortisol with stress increases. You look at saliva before a stress event, you look at it after, and the stress event should increase the cortisol. That's the normal response. What happens with the cocaine-exposed kids is the exact opposite. So the number of kids who show a negative response, a blunted response, many more of those are in the cocaine-exposed kids. And here's our friend violence again. So it's not just the cocaine, it's the combination of being exposed to violence and prenatal cocaine exposure together put all of this stress on the stress system, wears it out, and the system is worn out, which is why you get this blunted or reduced response. And this cortisol blunting, when the cortisol is not released the way it's supposed to, it is related to a whole bunch of stuff. Again, ODD, conduct disorder, maltreatment, all of these things that are severe stressors, severe adversity over time. Allostatic load is what it's referred to, where you're just wearing out the ability of the body to respond. If you go back and look at our disinhibition measure, again, no great surprise. The kids who showed the blunted cortisol response were the kids who had the most, the highest disinhibition or most problems with disinhibitory control. So this is a summary. This is a summary of what I think it is that we're seeing now, and in terms of our current understanding of how drugs operate in the context of environmental adversity, and again, thinking of drugs from the point of view of its impact on the stress system, where you have prenatal exposure, so that in utero the stress system gets disrupted. Okay, you get let's say an altered level of responsiveness, so that the stress system is the threshold is either too low or too high. The baby's born, the kid's exposed to community or domestic violence, and then that continued activation results in downregulation or a blunted cortisol response, poor inhibitory control, which is our neurobehavioral disinhibition, and leading to substance use, and the cycle kind of repeats itself. So, summary: what we seem to be seeing is that early experiences can affect adult health, and including mental health, in at least two ways. One would be by the biological embedding of insults during sensitive periods, and the second would be by accumulating damage over time due to chronic stress. So obviously in our case, the biological embedding of insults is drugs, and the accumulating damage has to do with environmental adversity, child abuse, domestic violence. So that, parental substance use and family violence are major risk factors that are included under the description of toxic stress. This was written up by the National Scientific Council on the Developing Child, that refers to chronic activation of the HPA and related stress response systems, resulting in stress-related disorders. So think about drugs as creating stress-related disorders, and stress-related disorders including psychopathology, drug use, and other kinds of externalizing behavior. So, again, it's likely that the combination of what we saw, the biological embedding of cocaine and the cumulative stress, those two things resulted in this cortisol blunting, neurobehavioral disinhibition that led to adolescent substance use and risky sexual behavior. So that's the end, and this is just, I could put 200 people up here because this stuff involves, 2 different longitudinal studies and the epigenetic stuff we're doing and everything else. But, these are just some of the major players and the NIH people as well as the people at the different sites. I guess I have a few minutes for questions.
I actually have a few write-in questions, so I don't think we're going to open the lines because there are a few folks who have written in and haven't had their questions answered. So, a lot of people are seeing prescription drug use among pregnant women go up, and they're wondering if you know of any similar studies looking at prescription drug use.
I am shocked and embarrassed to tell you that, as far as I know, there is not one single NIH study that's doing any kind of longitudinal work like this on prescription drug use. I don't know why. I know people who have submitted grants and they haven't been funded, but I do not know of any that are going on, and it's awful. I mean, it's something that really needs to get done. And I can tell you, if this sequestering, kicks into effect on Friday or a Saturday, there may not be an NIH to do any of this stuff.
Right. And then going back to your longitudinal study on prenatal cocaine exposure, we had some questions about whether there were differences noted within that PCE group that were associated with later early onset of drug use. Specifically, they were thinking about ongoing substance use of the caregiver, child placement, so whether, at home or in foster care, that kind of thing, if that was extrapolated out.
Yeah. Well, I showed you there was one case where there was an effective child abuse. There were several cases where violence was involved. I mean, the way these analyses are done is that you identify all of these potential environmental factors, right? So caregiver changes, maternal psychopathology. We have probably a list of 10 or 20 of them, and then when you run these analyses, you see which are the ones that are actually statistically significant. Which of them are contributing. So we looked at all of those, and I showed you the ones that came out statistically. I mean, we have other papers and other kinds of analyses where some of these other factors come out. But in general, I would say if I look across all of the papers that we've done at these different ages, there is a consistent set that seem to come out that you see across many of the papers, and they are child abuse, domestic and community violence, number of foster care placements for caregiver changes, and maternal psychopathology. I mean, there are others that come out sometimes, but those are by far the most consistent across different papers.
Okay. A lot of folks have questions and they're just really interested in this topic. Many of them want to know about possible interventions, and I realize that wasn't the goal of this webinar. But I'm wondering if maybe you could email me any resources that you know of possible interventions with these kids, and I could then post it on our website and make it available to our participants.
Sure. I have to say, there are not very many interventions that are developed specifically for, cocaine exposed or methamphetamine exposed kids, and I think the reason, this is a long conversation, and you're right, it wasn't the point of this, but truthfully, because we see these kids all the time. Typically, what you do is you treat the symptoms. If a kid has ADHD, and you treat the ADHD. If the kid has conduct disorder, you treat the conduct disorder. And whether or not the kid has it because they were prenatally exposed or not, in a sense, it doesn't matter. For one thing, you would never know if it really was the cocaine or the methamphetamine that resulted in the ADHD, right?
Mm-hmm.
It's in the kid's history, and you might think because it's there, it's the causal agent. But that's not necessarily true. There are lots of kids who have ADHD with no prenatal drug exposure. So you don't know if that was the cause. And even if you did know, you wouldn't do anything different. You would still treat the ADHD or the
right.
the acting out behavior. So people are always looking for the magic bullet, that's going to work for this particular kind of kid, and it's not there, and I think it's not necessary. I think the critical thing is that you have to appreciate the familial and environmental complexity in which these kids live, and you can't just treat the kid. You've really got to treat the whatever kind of environment the kid is living in, the people that the kid's living. It becomes a real family issue, and I think you need to go at it from that, what we would call infant mental health, the child mental health perspective, where you're seeing the problem in the context of a family.
Great. Well, as we mentioned, these slides and this presentation will be available on our website. Generally, it's up within 2 weeks. Our website is aia.berkeley.edu, and feel free to share this presentation with colleagues who may be interested. Dr. Lester, thank you so much. This was wonderful. Just the level of feedback and participation we have from the group really shows how interested people are in this topic. So we'd love to have the references to your studies as well that we can post on the site. If anybody's interested, we are having a 2-day skills building training here in Berkeley in June on clinical strategies to break intergenerational cycles of trauma, building on parents' strengths, with Dr. Sharon Melnick. And again, you can find more information on our website, aia.berkeley.edu. Also, before you sign off, if you could just take a brief moment to participate in our survey. We love to hear how you think it went. We really do take your feedback into consideration. If you're listening with a group of people, we'd love to have each of you click on this link, and that really does inform our webinar series. If you registered for CEUs, you can log in now at AATBS and take their exam so you can receive your CEUs. If you haven't registered yet, you can still register after the fact. So again, Dr. Lester, thank you, and thank you to our participants, and I wish you all a wonderful day.
Thank you. It's my pleasure.